SNMMI's Patient Advocacy Advisory Board. Thank you so much.
Thanks, Linda. Thank you, everybody, for attending and for coming. I'm, as Linda said, I'm Mike Crosby. I'm the chairman of the new chairman of the Patient Advisory or Advocacy Advisory Board at SNMMI. I'm also a three-time prostate cancer survivor. So if you have any questions, you know, we're still standing after many years. One of the things that we're here to do is actually to share stories. So if you make some new friends and talk about your pathway through your own journey, just remember it can help everybody else to get through these things. So to get through some of the things here, we want to let everybody know that we are going to be live streaming this. It's now working and doing quite well. We'll be doing the educational sessions on YouTube, and these will be available afterwards as well, so in case you miss it, you can go back and see it. We also want to welcome our virtual attendees from around the world. I mean, not just Canada and United States, but Australia, Argentina, Algeria, Belgium, Greece, Nepal, New Zealand, Mexico, and Switzerland. So it's a truly diverse crowd here with us today. This is the 13th year that SNMMI and the Patient Advocacy Advisory Board has held this event in conjunction with the annual SNMMI conference, which you'll get to experience later this afternoon. Many of the board members are here today on the Patient Advocacy Advisory Board. I'd please like to stand and acknowledge those board members. Thank you. Thank you for your service. Now, just a couple of admin notes, as Linda touched on first, but a couple of things. If you drove here today and you parked, you can have that parking validated, and if you haven't gotten your validation ticket, see Karen Bro, who's sitting out at the desk out there. What else have we got? Second is that, again, these are being taped, and for all of you online, on the YouTube channel, if you ask a question in the chat, that means that you're giving consent to being recorded. So just a little small point, the legalities of things. And if you need restrooms, they're out the door to the left and then up the steps and make another left, and if you need to take a break there. Real quick, I want to go through the schedule for today. In just a few minutes, we're going to begin the general session to talk about what is nuclear medicine, and we're going to touch on clinical trials as well. So really, I think two big questions, and hopefully it'll clear up a lot of issues. After that, we're going to have lunch. Lunch will be held here. It'll be a buffet luncheon, and please help yourself. And then we're going to have two speakers during lunchtime. First is Miranda Edwards, who will share her really fascinating story about her patient experience in having a rare disease. And really, the big thing to take away from her story is how nuclear medicine has helped her through that disease state. Then we're going to have a speaker for all of you Canadian hockey fans. I think you'll remember this guy, Mr. Wendell Clark from the Toronto Maple Leafs. And we're going to talk about what he's doing to raise awareness for not just prostate cancer, but also cancer in general. And doing a pretty amazing job, and also being supported by the NHL. So it's an exciting time of the season, so all you fans can bring your jerseys out or wave your flags, whatever you want to do. Then after lunch, we're going to head down to the exhibit hall. It's a bit of a hike, but we'll go down to the exhibit hall to learn about some of the technologies right now that are that make nuclear medicine scans and treatments possible. And we'll check out the SNMMI poster hall as well. There are going to be some researchers and physicians there that are doing the research that are, you know, saving us all, and really some exciting news going on in this field of nuclear medicine. And during that part of the session, please bring your questions and make it an open discussion. They're all willing to talk about what all the research is, the benefits it's going to have, and you know, you never know, you may run into somebody that's doing a trial that you can talk about. Then we're going to come back here after a full day of learning, right? And you're going to have a chance to relax at a little reception here. So we'll have a reception, get to know everybody, and share your stories, and make some new friends. Then after that, we're going to, you're all invited to SNMMI's Mars Shot reception, which is back in the reception hall. And Mars Shot is a program where SNMMI is trying to raise $100 million for research into nuclear medicine and how it applies to saving lives. So it's something for all of us to participate in, and it'll be a nice reception. And you can still, you can go talk to more of the vendors and some of the researchers out there. Now, any questions about the schedule? If you need assistance getting all the way over to the other hall, let us know, and we'll make arrangements to get you over there. But before I introduce our first speaker, I really want to say thanks to the sponsors for the program who really make this possible. First is Telix Pharmaceuticals, Blue Earth Diagnostics, GE Healthcare, ITM, and Lantheus Medical Imaging. Without their sponsor support, this wouldn't be possible, so it's a great thanks, and thanks for their generosity. Okay, so let's get started. So our first speaker this morning is Dr. John Sunderland. John's from the University of Iowa. Dr. Sunderland serves as the director of the university's PET Imaging Center, is an associate professor of radiology, radiation oncology, and physics and astronomy. In his spare time, he just does more studying. So, he's also chair of SNMMI's Research and Discovery Domain. And Dr. Sunderland is going to share an overview of nuclear medicine and molecular imaging, radiation safety, and how it applies to all of us. So let's give a warm welcome to Dr. Sunderland. John?
Yeah, thank you. In my spare time, I do CrossFit. I don't study. All right, so a couple things here. First of all, I'm fighting both allergies and asthma, so if you see me take a puff or something, excuse it, and I'll try not to be coughing my way through the presentation. Secondly, you've got a really exciting day ahead of you, and you may or may not appreciate how exciting it is. I mean, this field is exploding, and while pharma in general is struggling right now in a lot of phases in this radiopharmaceutical therapy space and imaging space, it's exploding. It's the one expanding bright area. Right now, large pharma is buying up these small pharma companies like crazy, because this is pretty much where the future is. So what you're seeing now, you're kind of in on the ground floor of what's coming. We've been doing this for a number of years, but this whole therapy space is getting really exciting. So, and I'm just kind of the setup man. The exciting stuff is kind of coming down the road. My job is just to try and kind of do some basic education. I'm a physicist by training, so you're going to get a little bit of chemistry, a little bit of physics, but it's not that much. But you kind of need to understand that in order to have an understanding of how the therapy works, how the imaging works, where the benefits all come from. So, with that, we'll start. So, this is kind of the roadmap of where we're going. First, we're going to do a little prelude, the difference between kind of the standard anatomic imaging they might be familiar with and actual the nuclear medicine and molecular imaging. We're going to talk about how nuclear medicine and molecular imaging works. We're going to talk about what it's used for, both diagnostic and therapeutic applications. I've been doing this probably for 5 or 6 or 7 years. There never used to be a therapeutic application. Now it's a big deal. And then some radiation safety considerations, because some people are understandably concerned about the radiation aspects associated, really both with the diagnostic and the therapeutic, and they're really, frankly, very different. So we'll just do a little bit of that, just kind of a basic overview. So what you see here on the left-hand side is kind of the anatomical imaging that you're kind of used to. I'm not going to go over these in detail, but there's MRI, which has exquisite soft tissue contrast. We can look at blood vessels like angiography you see in the kind of the top right one, kind of go down, ultrasound, you're all familiar with ultrasound, and then the most diagnostic CT scan I've ever seen, because you can see the lung cancer, you can see the emphysema, and you can see the pack of cigarettes in the upper right-hand corner of the scan. But on the right-hand side, you can actually see the molecular imaging. So in molecular imaging, we're not imaging the anatomy. We're imaging biochemistry. We're imaging the function of the body. We're imaging the nature of the disease. So, for example, in the upper right-hand corner, that's PET and Alzheimer's disease. That's imaging the amyloid plaques in the brain. On the right-hand side, there's a patient who has Alzheimer's disease. All that red there, that's amyloid plaques. We inject a radioactive compound that binds to that amyloid so it kind of lights up. On the left-hand side, that other color picture is kind of normal. So there's the normal. So we can image and diagnose Alzheimer's disease because of what we inject, because of the radiopharmaceutical. We can look at perfusion, what parts of the heart are actually getting blood and what aren't getting blood. That's the lower right. And on the left-hand side, and we'll see several of these because they're kind of interesting and kind of easy to digest, we're imaging glucose metabolism in the body. That's what that kind of up and down one is. You can see how it's all dark in the brain. That's because your brain utilizes glucose as its primary source of energy, really its only source of energy. So there's a lot of activity. So dark means a lot of glucose metabolism. You can also you can see it in the heart a little bit. You can see the bladder. The bladder's not activity. That's the kidney recognizes this radioactive sugar as an imposter, and so it actually filters it out. So you kind of see the kidneys and the bladder. But then, most importantly, you can see that tumor, because tumors almost always use glucose like crazy as their energy source in order to divide and grow. And so this is a really good way to see where cancer is, almost regardless of the cancer. Some it's not particularly good at, but most it is. So now let's just go through what is nuclear medicine and how does it work. So in all cases in a nuclear medicine study, we're administering, usually injecting, but sometimes we breathe it in or something like that, a tiny quantity of a drug that has a radioactive atom attached. So that's a sugar molecule over on the left-hand side. That's a glucose molecule. And we take off an oxygen and a hydrogen, and we replace it with a radioactive fluorine-18. So there's radiochemists who do this particular step. And it's not just one molecule. There's like lots of them that we all label. So we got a little tag on it so we can follow it around the body, right? That's what's ultimately going to happen. What we inject is a really tiny, tiny amount, physical quantity. When we inject this radioactive sugar, it's about a millionth of a single grain of sugar. So the mass quantity is tiny, tiny, tiny. Okay? But there's enough radioactivity in it that we can actually detect it. And we detect it from the gamma rays, and we're going to be getting into that in a minute. But we're always, and what we see in the image depends on the molecule, right? So now, a little bit of chemistry, but not a lot. You've all taken chemistry class at some point in time. You're all familiar with the periodic table, right? All the elements. I consider this a kind of a shallow two-dimensional representation of a three-dimensional space, because each of these elements has different isotopes. And what I mean by that is we're going to look at carbon. Carbon-6, we all have carbon in our body. Okay? Carbon is anything that has 6 protons in the nucleus, by definition, but there's protons and neutrons that are in there. And carbon-12 has 6 protons and 6 neutrons. That's 6 plus 6, right? Most of the carbon in our body, more than 99%, is carbon-12. But there's also another stable isotope of carbon, which is carbon-13. That has 6 protons, so it's still carbon, but it has 7 neutrons. But that's stable too. But if somehow you get an extra neutron in there, and there's now 7 neutrons and 6 protons, that makes the nucleus fundamentally unstable. And so it's radioactive. So when there's not the right balance between protons and neutrons, the nucleus is unstable, and it decays by radioactive decay. And there's a couple different kinds of radioactive decay, not a lot. We're going to get into that in a minute. But that particular one, if it has too many neutrons, it decays by beta decay. It actually emits an electron from the nucleus. Okay? We can actually go the other way. If it has too few neutrons, it's got 6 protons but 5 neutrons, now you have a proton-rich nucleus. That one's not in balance. You've got to get rid of that extra positive charge, and so the nucleus also decays. This time, it emits a positron. That's a positively charged electron. You may or may not have heard of that. It is an antimatter electron. Yes, there's such a thing as antimatter, and a positron is antimatter. And so in our PET clinic, we use this all the time. We goof around with antimatter, and it's fun, and it's really useful. And so we can take that carbon-11, we can put it in this case onto a choline molecule, and we can use it to detect prostate cancer. This is one of the first prostate cancer agents that was approved. Mayo Clinic actually used that, and they're still using it today. You can see that kind of little dot down in the pelvis there, right? And that's a PET scan that we're seeing there. Okay, so Here's just some examples of some radiopharmaceuticals that are out there that are approved, FDA approved, and they're out there for different cancers. They have different specific biochemistries. On the far left, we have fluorestradiol. So in breast cancers that are estrogen receptor positive, this binds to those cancers that are estrogen positive. If you have an estrogen positive cancer, then you're eligible for certain really effective therapies that have really low side effects. But if it's not, then you have to go to something different. So this helps guide therapy. Or in the middle, you have PyL or Pylarify for prostate cancer, very specific. It binds to prostate-specific membrane antigen, which is expressed on prostate cancer cells almost exclusively. And so you can, this has revolutionized the staging and the treatment of prostate cancer, approved just a couple years ago. On the right-hand side, we have copper-64 dotatate for somatostatin receptors, and we'll talk about that in a minute. That's for neuroendocrine tumors. But that's, right now, there's 17 approved PET agents right now on the market right now. So this is just a handful. Okay, so now a little bit of physics, a little bit of radioactive decay, but not too much, because there's really only 3 kinds of radioactive decay, right? Too many neutrons, it emits an electron from the nucleus. That electron travels about 1 millimeter in your body. So if you have these drugs in your body, that electron's going to travel only about 1 millimeter. If there's too many protons in the nucleus, it decays by positron decay. That's just a positively charged electron, antimatter, which is ejected from the nucleus. It too travels on the order of 1 millimeter. This one's a little bit different, though, because it's an antimatter electron. That antimatter electron travels only about 1 millimeter, but then it runs into an electron. That's the whole matter-antimatter thing. That's the whole Star Trek matter-antimatter, engine. When matter meets antimatter, they annihilate. They disappear. An electron has mass, a positron has mass, but when they get together, they annihilate and disappear. No more mass. That whole mass can neither be created nor destroyed. Not true. It happens here all the time. But what you get in its place are 2 gamma rays that go off in opposite directions. And the energy of those gamma rays is equal to the original mass of that electron and that positron, according to that whole E equals mc squared thing. E is energy, m is the mass. Take that mass, multiply the speed of light squared, you get 511 kiloelectron volts. So that's where this comes from. So the mass disappears, but you get energy in its stead. And it's these gamma rays that the PET scanner detects. Okay? And on the right side, and I always glanced over this because who really cares about alpha decay, but alpha decay is all the hot rage in therapeutics right now. Alpha is when we have large molecules, they're unstable, they will frequently emit alpha particles. That's 2 protons and 2 neutrons together. They travel only about 2 or 3 cell diameters, but they cause all sorts of damage along their route. That's bad from an imaging standpoint, but it's good if it's attached to cancer cells, right? Because it's blasting through like a big cannonball, breaking DNA strands and killing. So anyway, but those are the only 3 we got. So that's kind of easy. When they decay, by alpha decay or beta decay or positron decay, they often give off gamma rays. Because the particles only travel a short distance, our scanners don't detect those. They detect the gamma rays that are coming off, right? So if it's PET scanner, we're detecting those 511 keV. If it's a SPECT scanner or a single photon, then we're just detecting the single gamma rays that come off of different energies. Okay? Because those ones, they have enough energy, they go through the body. When you go to the floor, you're probably going to see some of these. PET scanners on the top. We do that when they're positron emitters. There's a SPECT scanner on the bottom. That's when they're single photon emitters. They decay by beta decay, and sometimes alpha decay. We'll use those. Planar gamma cameras, you'll see a few of those on the floor if you're looking, but the reality is, if you ask me, those are going the way of the dinosaur. It's kind of old technology. They were being replaced by the SPECT systems, which give you kind of nice cross-sectional images, like kind of CT scans and stuff. So, these days it's going to be PET and SPECT. So if you're having an imaging study, this is typically what happens. So if you look on the left, the drug is usually administered just via IV, usually in the arm, not always. So that's what's on the far left. What you see in the second panel, and it'll loop around, so we'll get there. This is an injection of fluorodeoxyglucose. That's the radioactive glucose. Interestingly, for illustrative purposes, this was actually injected in the foot. And when it starts at the beginning, you'll actually see it kind of go up the vein of the foot. It then ultimately goes to the heart, which then goes to the lungs, goes back to the heart oxygenated, and then it goes to the rest of the body. And what you should see here as it goes along is you'll see that in the beginning, there's no activity really anywhere, but certainly not in the brain. The brain is the thing that uses primarily most of the glucose. And you can see it leaves the bloodstream, it goes into the tissue to where it's actually being metabolized. And this is just showing, this is now 2 minutes in, this is kind of 3 minutes in. You see the brain kind of lighting up? Okay, that's the brain utilizing that glucose. Usually we inject and we wait about an hour. And by that time, you get a map like you see over on the right. The scan itself takes on the order of 15, 20 minutes or so. You can see there's a little bit of metabolism in the liver, which is that big kind of organ in the middle. You see a lot of activity in the brain. You see a lot of activity in the heart. It's a muscle, right? So it's pumping and it needs energy. This is normal. This is not a cancer patient, so we don't see any cancer here. But that's the procedure. You inject, you wait, you scan. It's painless. And fairly rapid. What is nuclear medicine used for? What are the diagnostic applications? And what are our therapeutic applications? That's where we're going next. Okay, these are, once again, just some examples. So cancer detection, you can see the FDG on the left. We use it in cardiology, primarily but not exclusively, looking at blood flow to the heart to see where there's stenosis and if somebody's having a heart attack or ischemia, they're having chest pain, you can see what parts of the heart are not getting blood at the microvessel level, right? This is at the capillary level. We're seeing where blood is actually getting to different parts of the tissue. I have this lung imaging only because on the left-hand side, this is imaged with xenon-133, which is inhaled. So that's one of the few that's actually inhaled as opposed to injected. It's usually a two-part study where you inhale xenon-133, but then you get injected to look at kind of where the blood is going in the lung, and you kind of use the information from those two. I showed an Alzheimer's disease image before, but there's all sorts of other brain applications, including Parkinson's disease, and there's radiopharmaceuticals, both PET, positron emission tomography, in the form of fluorodopa, and SPECT in the form of DAT scan that are out there to help diagnose Parkinson's disease. And these are also used in research, because you can image the development, how much of the structure is being impacted over time. You can follow drug development. You can use drug development to see if you can slow the progression of Parkinson's disease, for example, or slow Alzheimer's disease. We'll talk about that again in a minute. So here's just another few specific examples. This is gallium-68 dotatate. So I always have kind of the molecule over on the left. I have a sample image next. This is used in neuroendocrine tumors. It's been around. It was one of the first non-FDG cancer agents that was approved. It's used for staging and monitoring response to therapy. It's not quite ubiquitous. It's not everywhere because that gallium-68, that's that radioactive label, it has a half-life of about an hour. That doesn't mean it goes away entirely in an hour. It just means half of it disappears in an hour. So when we make the drug, if the patient is 4 hours from now, after 1 hour you have half, after 2 hours you have a quarter, after 3 hours you have 8. You have to make 16 times as much in order to, so we have to make it just beforehand. And so you can't make it and then put it in a car and send it 3 or 4 hours away because most of it's gone by then, right? So you end up having kind of a radius around which you can actually treat or inject in patients so they can have that diagnostic scan. That's why the whole U.S. is not kind of covered there. We have those radii around where we're actually doing these studies. But a company came up with copper-64 dotatate. That's the same molecule, right? But instead of gallium-68, they put a copper 64 on it. That's another positron emitter, so you can use PET scans for it. It has a half-life of 12 hours. So they can make it in St. Louis, and they can send it anywhere in the country 12 hours, 24 hours later, because they only have to make twice as much or 4 times as much. And they make a lot of it, and they ship it all over the country. So here we have now coverage for neuroendocrine tumors pretty much around the country if you wanted to go that route as well. So there are options out there. And there's an example of a scan, all the same, otherwise it's all the same and all approved and reimbursed. Here's some FES, this is the fluoroestradiol for breast cancer, and here we're seeing the estrogen positive receptor tumors, which are over there on the left that you can see. We usually compare that with an FDG scan, which shows all of the tumors, because sometimes these tumors are heterogeneous, so some of them express estrogen, some of them don't. If that's the case, you may not want one of these estrogen therapies out there because you're only going to be treating some of them. But if all of them are estrogen receptor positive, then you're eligible and you can get the right treatment for the right disease. It'll be very effective without many side effects. Here's the prostate cancer agent, one of several now, and there's a lot more coming down. These are in clinical trials, late phase clinical trials. This is one of them. Pretty much it's amazingly sensitive at detecting even small lesions down to 3 millimeters in size, which is crazy. And this helps you understand where the disease is. It helps you follow the treatment of the disease, et cetera. It's approved. It's widely available. And we try and find recurrence with this. So if the PSA starts going up, after treatment, where's the disease? This is when you get this scan, and you can say, oh, there it is. And if it's just one place, you can do radiation therapy, or you can do even surgery. If it's all over, then you have to do a different kind of therapy, which we'll talk about in a minute or two. This is just another agent for prostate cancer that was more recently approved, Pasluma. Same thing. A little bit about brain imaging. For more than a decade, there have been 3 amyloid agents that have been FDA approved for the detection and diagnosis of Alzheimer's disease, and almost nobody used them. Nobody used them because Medicare refused to pay for them. They refused to pay for them because there was no treatment for Alzheimer's disease. So what if you have it? You can't treat it, so we're not going to go there. Now, there's some Alzheimer's disease treatments coming out. And so they recently said, okay, we're going to start paying for these scans. That just recently came. They have their non-coverage decision was reversed. The problem is now that they're paying sites like the University of Iowa $1,300 to perform one of these scans. The drug itself costs about $5,000. So if we do these scans, we do them at a loss. Whether the patient is or isn't responsible for that for that addition, it just depends on their age and their insurance and all this other stuff. But it's not optimal right now. This is not that big a deal because the drugs aren't that effective, but there are some that are coming along that look better. And so this is a keep watching this space, and this is the kind of the dance that we have to do in this area. I'm going to spend a little bit of time on therapeutic applications here. I love to speak to an audience who probably has actually seen The Graduate, right? So in the Graduate, during the graduation party, a family friend comes up, puts his arm around Ben, and he says, I just have one word to say, just one word, plastics. Well, if this were done in 2024, not 1997, it probably would have been just 2 words, radiopharmaceutical therapy, because this is a big deal. It's a big deal right now. Companies are really scrambling in this space because it's looking potentially so promising. So one thing we do know about radiation is at very large doses, radiations can kill cells, or at the very least damage the DNA so they can't grow like cancer cells. And that's the foundation of external beam radiation therapy, right? In external beam radiation therapy, in that kind of central area, that's where the tumor is. And the way that works is you give a sublethal dose of radiation that, like X-rays, that kind of blast through the patient, and then you do it from another angle, and you go through, and where they overlap, now that tumor's got twice as much dose, right? And then you do it from another angle, and the tumor now has 3 times as much dose, and that's enough, hopefully, to kill the tumor cells. But the cells around it, they got a lot of radiation, but hopefully not toxic levels. And that's the way they play, and I don't want to say hopefully, they actually measure the radiation dose very carefully so that it causes little extra damage as possible while still getting it. And that's great if you know where the cancer is and it's localized. But that's not always the case. Sometimes you have a situation like this, and this is a patient with neuroendocrine tumors, with a huge tumor burden, right? You can't aim external beam radiation therapy at that. It's everywhere, right? But those scans I showed you before get you thinking. We're injecting a little bit of radioactivity, and it's very specifically going to the tumors. It's not going to much else, right? It is going to other places, but not much else. Now we're putting gallium-68 on that has a half-life of like an hour. And so we inject it, and it emits that beta particle, gives you a little bit of radiation dose, and it just goes away. 4 or 5 hours later, it's almost gone. But what if we took that same molecule, and we didn't put that gallium-68 on it? We put lutetium-177, for example. Lutetium-177 emits a beta particle that travels about 1 millimeter or so. And it's got a half-life of 7 days. So you inject that into the patient, and it emits gamma rays too, so we can image it, so you can see it there. And it goes exactly where we expect it to. So this is not the PET scan anymore with gallium. This is actually a planar scan that you can see, but it all goes there. And what's happening is those molecules are bound to the cancer cells, and they're emitting these beta particles, these little bullets, like little machine guns. And it stays in there with a half-life of a week. It stays there for a couple weeks, continually shooting little bullets at these cancer cells, to the point where if you hit enough of them, it's going to kill the cancer cells. And then you can get a result like that, which is crazy. Now, this is what we call a super responder. This doesn't happen all the time, so don't expect it to happen all the time. But this is an almost complete response to one of these therapies, and frankly, it's amazing, right? And this is what's captured the attention of everybody. More often than not, you get stable disease, you get some reduction in the bulk or whatever. I would argue that we'll see, and I'll show you in a minute, I think most of us in the field agree, in general, we're under-treating patients. The reason we don't get enough responses like this is we're not giving enough radioactivity to actually kill the cells, and we probably could give more, and I'll talk about that in a minute. The whole use of a diagnostic nuclear imaging, which is what we're doing on the left side with that gallium scan, that's the diagnostic to qualify the patients, followed by a therapeutic dose, is called theranostic. So you may have heard that term, or you may hear it moving forward. And it's called, and the therapeutic part is called targeted radionuclide therapy. There's a lot of, we still haven't come up with a standard approach. So lutetium-177 dotatate was approved in 2018 by a small to mid-sized company. And you can see here another successful. They were bought by Novartis for $4 billion. And all the other pharma companies ended up saying, what do they know that we don't? And so at that point in time, people started looking into it, and now there's kind of a feeding frenzy with large pharma not building up their own programs, they're buying small pharma, which means now there are about 150 companies right now in this space, startup, because they want that $4 billion, right? So it's fascinating space to watch. This got approved because of much better kind of survival, progression-free survival curves over the standard of care. So the people who got Lutathera is on the top. People who got kind of the standard of care is down below. Just going through it quickly. This is now moving to prostate cancer. Just so you understand the format here. In each case, A, B, C, D, E, and F, right? You have the before scan, and what's highlighted in red is where the cancer is before the treatment with lutetium-177 PSMA. So we're not dealing with Lutathera anymore, we're dealing with PSMA, prostate cancer agent, before and after. So the first one had functionally complete response, and then if you go down to the bottom, you can see not quite complete, but really remarkable responses. Once again, pretty much best case, but it also shows you that sometimes you don't get it all, and sometimes you don't get any. Sometimes they're non-responders completely. But right now, from an approved standpoint, we have neuroendocrine tumors and we have prostate cancer. Okay, that's where we are today. So, sorry, here's the kind of the survival curves once again showing the benefit. So this is, I just want to show you where we are right now in the therapy space right now. So, and this is a year old, and so this is outdated, and there's probably 10 more. We have 9 in phase 3 trials. That means they're trying to get approval. A lot of those are actually still in the prostate cancer space or the neuroendocrine tumor space, so may not be expanding the diseases being treated in phase 3, although possibly. Some of them are in earlier phases. Most of these are being treated in the later phase, so we're trying to get more patients to qualify. 15 in phase 2, 32 phase 1, and 114 in development. This is probably double by now, just after probably a year and a half or so. We're not just talking about prostate cancer and neuroendocrine tumors anymore. There's an array of cancers out there. That's what all these smaller companies are aiming at. We want a new market. We want to be the only one in this space. So there's a large array and this is all information from clinicaltrials.gov that we garnered about a year ago, and it's a really rapidly evolving space. Just looking at an estimation of patient enrollment here, there's our phase 1 to phase 3 that I showed you before. The phase 3 trials that are at the top, they are treating a lot of patients, right? So that's why you see there's a lot of blue on the right side, estimated patient enrollment. We're talking about, as of last year, probably 6,500 people in clinical trials in the therapeutic space, which is a lot. And once again, it's probably 1,000 or 2,000 more by now. So there's a lot of opportunity in this space to participate in these trials. Quickly getting into some radiation safety considerations here. Sorry. I just want to bring up that there's 2 kinds of radiation risk that I want to talk about. The first is the stochastic risk, which is it's the probability that downstream you might get a potential cancer. This is what we worry about in diagnostic nuclear medicine. We're giving a small amount of radiation. The probability of it actually causing cancer is there. We don't really know what it is. It's been estimated, and I kind of have it here. But when I say this is statistically based, okay? So, and this is like the odds of getting struck by lightning in your lifetime, they say is 1 in 15,000. That seems like a pretty high probability to me. But nonetheless, I've seen a couple of sources, and it doesn't seem to be too much off. Lung cancer, if you smoke a pack a day, you got about a 1 in 4 chance of getting lung cancer. The estimates suggest that if you got a single PET or a single CT scan, it may present a risk of developing cancer from the radiation of about 1 in 2,000 over about a decade. Most patients that get PET scans are on the cancer side. They're usually late stage. You usually don't have to worry about 10 years down the road because you're worried about 1 year down the road. And the probability is still very low. It's one of the reasons, though, that we're particularly careful with children, because they're not just living, they don't potentially living just 10 years, and that it's more important to look not 10 years, but 20 years, but 30 and 40 years down the road. So we take particular precautions when it comes to them. So that's this risk on the diagnostic side. When we're doing therapy, we're worried about something different. We're worried about deterministic effects. That is, we're trying to kill cancer cells, and we are inevitably also going to be killing some healthy cells. And we can actually cause damage to tissue. We can actually have bad things happen. But if anybody's had chemotherapy or external beam radiation therapy, this is what happens with treatment. And so you have side effects, and what are the side effects, and how frequent are those side effects? And that's what we look for here. You could have kidney damage. You could have damage to your bone marrow, which has to do with your immune system, and platelets, which has to do with blood clotting and all sorts of other things. We have to watch out for that, just like they do in chemotherapy, in immunotherapy, and whatever. So, and those are real, and we do have to watch those. I personally don't worry much about the diagnostic doses. I pay very close attention when I'm doing therapies, okay? So going to the low levels, which is kind of diagnostic, there's radiation everywhere. There's radiation in these walls. There's radiation in your house. There's radiation in your body, naturally occurring, long-lived stuff that's just hanging out there. There's radon. And so that's, and we all get, and this is kind of nice, typically, and I'm going to use the millirem as our unit, we all get about 1 millirem of radiation dose every day. So you just kind of keep that in the back of your mind, right? So we get that, that's just natural, unavoidable, and frankly not that big a deal. I'm actually going to start at the bottom. So in the diagnostic range, that's the 10 to 1500 millirem, so that's a lot more than background, right? That's what we get for diagnostic procedures, CT scans, PET scans, those sorts of things. The people that work in my PET facility, they can get up to 5,000 millirem per year, and most of them get about 1,000 millirem per year, okay, for 30, 40 years. And they have that limit because the government says that's safe, because that's about an order of magnitude lower than we've ever seen any real radiation effects. So during the course of a year, your technologist is probably getting the same dose that you might be giving a patient in that one scan, just to try and kind of balance that, okay? 50,000 millirem to the whole body at once can slightly increase the downstream risk of cancer that they've actually seen in a really statistically significant way. 500,000 millirem will kill you. About a 50-50 shot of killing you. Okay? So that's just kind of the range that's there. The diagnostic procedures are really down on the lower end. Okay? But sometimes people get a lot of PET scans and a lot of CT scans, and that can add up. So this is just a list. Chest x-rays are nothing. They're 10 millirem, that's like 10 days. But when we get down to PET scans, you're on the order of 1,500 millirem. Background radiation in Denver at higher altitude gives you about 1,000. It's about 3 times the background rate than you kind of have here in Toronto. So anyway, that's just kind of what we're exposed to. How much radiation would be considered too much for a nuclear medicine diagnostic test? Well, the answer really is more than you need. And we pay close attention to this. But in virtually all cases, the benefit of the clinical imaging study far outweighs the risk. The risk is low. We don't do these scans for fun. We do them for a reason. And particularly in these nuclear medicine scans, a lot of them are life-threatening. You're trying to make a decision on how you want to move forward. You want accurate information. Each imaging procedure takes a certain amount of radiation to appropriately image. This is well studied. We know how much to give. We know what's too little. Using too much leads to unnecessary radiation dose to the patient. You can also go the other way. You can give too little. Because if you give too little, the scan quality is going to suck. And if the scan quality suck, your physician's not going to be able to make a good read, and they may miss that cancer that you're trying to follow. So there's a window here that we actually try to look at. We pay attention to it, we need to look at. Each imaging procedure is optimized for the medical question being used to the patient. It's just the way we perform medicine in the area. Just one slide on radiation safety and therapy. This is the deterministic effects. So this is information that's from the labels. So the FDA requires a label for the drug, for Lutathera and for Pluvicto. And if you look in there, when they do these large clinical trials with hundreds of people, they're required to report the adverse events. So there's grade 1, grade 2, which are kind of annoyances. Grade 3 and grade 4 are kind of serious. So I'm going to pay attention to the grade 3, grade 4 kind of events that you have here. So the organs at risk from this large amount of radiation are in particular the kidney and the bone marrow. And so here we're looking at Lutathera. In those clinical trials, there were serious side effects on the order of 2 to 5% for the kidney. That's not to say that that we ignore that. You don't want 2 to 5%. Marotoxicity also in the range of 2 to 3% of the patients actually had serious marotoxicity, which can be life-threatening. There were no deaths, but serious life. When you get into typical chemotherapy, those rates are on the order of 40%. I see that, you see that, and you say, ooh, it's safe. I say that, and I say we're under-treating patients. Because there's a lot of patients who don't get a lot of benefit from these from these therapies, and it's probably because we're not injecting enough therapeutic. And we can't just willy-nilly do that because it's the way what the label says, and we can't inject just willy more. But the clinical trials, in my opinion, they weren't optimized. We could tolerate higher risk to get higher benefit, my opinion. Okay? And we just had a big meeting with the FDA about this, because the FDA is very concerned about safety. And we're trying to come up with rules by which clinical trials can follow and go up to higher doses and not have the FDA get in the way and freak out because we actually have some serious side effects. Because they see serious side effects with all the other treatment drugs that are out there, right? So we're just trying to come to terms, and we're talking to FDA, and they're talking to us, because they see this as a big deal. We actually scheduled a meeting with them, which is hard to do. We asked them if we could come to the table and talk about this. And between that ask and actually that meeting was 3 months, which is crazy, because they were interested to talk to the community, because they want guidance. And so we had a good meeting of minds there. Pretty much the same thing with Pluvicto. Once again, I would argue under-treating, not a whole lot of side effects in this space. So very safe, effective, but once again, probably not as effective as it could be. And so we as a community need to do the clinical trials so we can be more effective at this. So with that, the imaging community in particular, and really I'd say the imaging and therapy community, but there's these dose optimization campaigns for imaging, Image Gently and Image Wisely for pediatric and adult imaging. Both campaigns promote using the appropriate procedure for the specific patient with a minimum radiation dose necessary to provide useful information. You can see that on the website, the SNMMI website. And I think that's my last slide. I would ask that people ask questions if they have them now, because I have to run to another meeting right after this. So please feel free. So. So this slide?
Yeah, that's the right one. So how I'm interpreting this is the radiotherapy that's currently available is not strong enough compared to what chemotherapy is doing.
Well, so that's my conclusion. It has to do with the way clinical trials are designed and the way they were actually moved forward with this. I think if they had been a little bit bolder in their clinical trial design, you would see higher complication factors, but we would also see better treatment results. This is all, it's a balancing act. So don't get me wrong, and it's not easy. But once you have a trial design, you get it going, you can't change it, is the problem. You have to bring that to completion. So there's a whole, and there are trials out there that are going higher doses, higher dose rates, et cetera. Hold on, here and then there.
Is there a correlation between grays and radiation and millirems?
Yeah.
Okay, so what's 1 gray in millirems?
So there are 1,000. So there are 100 millirem in a rad, or a rem. So you have to go up to that, and then there are 1,000 rads in a gray. And I hope I'm not, I'm just, so there's a lot of millirems in a gray, actually maybe 1,000 millirem in a gray. Yeah, no, that's wrong, sorry. Rem to rad, then rad to gray. So I'm a physicist, gray is a joule per kilogram, you know? But it's a big number. And that's why we don't use millirem when we're talking about therapies. So for perspective's sake, for perspective's sake, if you get on the order of 70 gray to a tumor, and it depends on the cancer, sometimes it's 30, that'll kill the cancer. And so that's kind of what you want to target. There's a lot of things that enter into it, but if you want to call it ballpark. The kidney dose that FDA wants us to worry about is about 23 gray, because that's where you start seeing potential damage to the kidney. It's actually higher than that. FDA was being conservative in that and will probably go up to about 30. Different organs have different tolerances. For bone marrow, it's on the order of 2 gray. So we have to be very careful about bone marrow. That's when we start seeing, but that varies considerably sometimes too, and it depends on the agent, not just the radionuclide. So 2 to marrow, on the order of 20 to the kidney, somewhere between 40 and 70, and maybe up to 100 to kill tumors. And those are and I should have mentioned this, but I didn't. We're using imaging to measure the radiation dose in these patients right now. So we should be able to be in a position where we can predict this. This is in the early phases, and there's a lot of scientific work. If you go over to that poster hall and you look for a dosimetry poster, because we're trying to get better at our imaging dosimetry so we can be predictive. And if we get really good at it, and that's really what I'm doing right now, is I'm trying to improve the quantitation in imaging so we can do dosimetry. So when you're doing the clinical trial, you can be adaptive. You can say, ooh, this patient's hardly getting any dose to the kidney. We can give this patient more therapeutic dose. Whereas this patient's actually getting a lot to the kidney for the same injected dose, so we have to be careful with them. That way you avoid the toxicities, you treat the patient up to what they can tolerate while still avoiding those safety concerns. And this is a huge area of research right now, and we're doing it in concert with industry, the scanner makers, so that we can do quantitative stuff there. We're trying to get the calculations nailed down and standardize the approach. So if I do the calculations and another physicist does the calculation, we get the same answer, because right now we're pretty far off. But scrambling in this space, because there's huge, huge potential to understand the radiation dose to organs, to keep that toxicity down, so we can keep those numbers down, but still give more therapy to those patients who can tolerate it and get better results. That's where we're going, but it's not where we are.
And I think we have time for just one last question over
Sorry.
here.
Hi, thank you very much for your presentation. It's been excellent. I've learned so much. And my name is Sarah Speary. I'm with the Canadian Neuroendocrine Tumor Society. I am a metastatic pheochromocytoma patient myself. I did not qualify for Lutathera because something was, I don't think I heard in your talk, just an observation, was due to the receptors on my tumors. Some were MIBG compatible, some were on Lutathera. So I am on the Welly-Reg trial at the moment, but I suspect this, once that stops working, this will be my future. My question is mainly for another patient at CNEX, where I think you've really just beautifully addressed it in the respect that he has gone through clinical trial for Lutathera, but he has had recurrence. And when I was at a previous conference, they did talk about dosing being sort of an unknown thing, and we're trying to find someone who will take him for a second sort of dosing or trial to help. And I don't know whether you're comfortable commenting on sort of second dosing. I think you did kind of address it.
So the answer is it is not uncommon for patients who are even in one therapy to then go to a second trial and another therapy when they failed on it. That was one of the interesting things that the FDA was interested in the conference, because this patient has already received some radiation dose to their organs. Now they're going to get more. How do we handle that? And that's an open question. And the answer to it is we need to collect the data. And so this is actually a very good patient, because if we actually collect the data, then we're going to, in a meaningful way, then we can supply it to FDA as safety data, whether it's good or bad. What's happening with a lot of these patients who do fail on, and I didn't get into it, but a lot of excitement over alpha emitting radionuclides. So instead of lutetium-177 that emits a beta particle, these alpha particles, they're potentially a lot more effective. We've seen that anecdotally, but these are almost all in phase 1, phase 2 trials. There's been one phase 3. So more often than not, for those who have failed on these earlier phase beta trials, what you look for is maybe an actinium-225 trial, and there are several of them out there. You'd have to look at where they are, and they could qualify. Sometimes they exclude patients who have had previous therapy, sometimes not. So you have to look at the inclusion criteria. But they are not categorically excluded. And that is an exciting area, but we don't have a lot of information on those yet. But that's where probably more than half of the trials are going right now, is in the alpha space, but they're all phase 1, phase 2. Okay. Great. Thank you. I appreciate your attention. Yeah.
Thank you, Dr. Sunderland. Thank you very much. And actually, I think this is a plant, because we're going to go right into our next topic is going to be talking about clinical trials. And I personally have a bent against the word trial, because I think that they ought to be clinical studies or data analysis or data acquisition programs or something, because we're still finding patients that scare away from the word trial, meaning that they think you're a guinea pig and you're just out there for nothing. But we're going to let our next guest talk about that whole issue. And I'm pleased to introduce Dr. Simran Singh. He's a medical oncologist and affiliate scientist at Sunnybrook Research Institute and assistant professor at the University of Toronto. In addition to his extensive training, he also has a master's degree in public health from Harvard. Dr. Singh is going to help us better understand these things called clinical studies and clinical trials. So, Dr. Singh.
Thank you very much, and actually, I couldn't have been better set up, because I'm going to talk exactly about that topic, about the word clinical trials and the sometimes the hesitancy that we have. And I really like your suggestion, so I'm going to steal that and maybe change this for the future. So I'm happy to talk today in general about clinical trials and what clinical trials are and what they mean and what this means in the patient journey and some of the common thoughts, but also some common misconceptions around this. These are my honorariums. So what is a clinical trial? And like I said, this was exactly what we're talking about. It means different things sometimes to different people. We think about research, we think about maybe experiments, we think about risk, but sometimes what we don't think about is the evidence that we're collecting that really change the way that we can treat disease, the possible steps we're taking, the scientific breakthroughs that we're taking. And a larger part is about being part of a larger community, about all collectively working together in this data acquisition to be able to move the field forward, in this case, in my instance, neuroendocrine malignancies, but in many different areas of cancer and even outside of cancer. So again, when we think about that word, clinical trials or clinical trials in general, so what are we thinking of? Well, some of the positive stuff, we think about opportunities. And one of the previous speakers, the question was about, what are the opportunities for accessing new treatments and accessing new ways of treatment? How do we get access to new drugs that maybe aren't yet approved? Clinical trials are often a mechanism for that. Help others down the road, and that's what I was talking about, the altruism or the community. It's amazing when I talk to my patients about really any sort of clinical trial, a big topic that comes up is, this is my way to contribute back. The extra visits I may have to make, the extra recording of data I may have to do. This is my way of being part of and helping people down the road who are facing what I'm facing today. And hope. Being part of breakthroughs, being part of cutting-edge treatments often inspires a fair amount of hope in people. The other side of it is it can install a number of fears. Am I a guinea pig? This is really big pharma making money on my back, and this is an area as well where people have a healthy degree of skepticism sometimes about trials. And it's important to know who's the sponsor of your trial, and we're going to talk about that. But often people think, well, this is just a money-making scheme, and I'm a guinea pig, and I don't want to be part of it. Risky. And there are risks involved in any clinical study, and I think it's important that people ask about them. But there's risks involved, as we heard from the previous speaker, of the therapies that we use now. It's no different that we accept a certain level of risk with any therapy that we use. And I'm not a test tube. I'm not the one who I want to be experimented on. And I think we need to think of this maybe in a larger picture as a forest about all the different pieces of investigation that are going on to help overall improve outcomes. So a couple of key components. Clinical trials, these are voluntary research studies. And I think this is what's very important, is that you can't be put on a clinical trial without your consent. And that consent can be withdrawn at any time. So you choose to participate in clinical trial. It's your choice. And if you choose to participate in a clinical trial, if at a certain point you decide you don't want to participate anymore or you've changed your mind, you are absolutely free to withdraw from the clinical trial. That is your autonomy that is maintained not only at the beginning, but throughout the pathway. So clinical trials are studies that are conducted in people to answer a specific question. Effectiveness, so how effective is a certain therapy? Safety, and we can have trials that really, the primary endpoint is to look at how safe they are. And I think it's important for people who participate in clinical trials to understand what is the goal of the overall clinical trial I'm appealing with, and many times it's both, and almost always it's both, but in some degrees we're looking, the early clinical trials will look more at safety, and the later clinical trials will look more at efficacy. Now, a clinical trial we think of as a drug, but it's not always a drug. Sometimes it's a device, sometimes it's a type of surgery or a place in the process of where surgery or a drug may be placed upon a patient's journey. Sometimes it's even a behavior. It's a change that we can make that potentially is looking at changing outcomes or safety or both. So there's some major principles of clinical trials that have to be maintained. And one is to protect the health of the people in the trial. That is paramount. So clinical trials are monitored. They're monitored blindly away from the investigators to ensure that there's no safety concerns to those people within the clinical trial. Even more important than finding the endpoint, and this is a point that often patients are surprised when I talk to them about this, even more important than the advance itself or finding if a drug works or not is to protect the safety of those in the trial. That is paramount. It must be well designed, i.e. it has to be designed in a way that is feasible, but also that looks at the safety of patients in the trial and tries to maximize the efficacy of the patient in the trial. There also has to be something called equipoise, which means that as a medical community, and that includes all of us in this room, we don't really understand or know which option is better, for example. If we think that a treatment's better, it's probably unethical to offer patients a suboptimal treatment. We really have to have something called equipoise, which indicates there is uncertainty in the community about which option is better. Clinical trials must be conducted by qualified, certified, and trained health professionals. And there is a huge amount of training that goes through in any area, in Canada, here, in the United States, wherever it might be. There's a huge amount of regulation around who can conduct a clinical trial, who can participate in a clinical trial. Anybody that speaks to you on a clinical trial has to have a certain level of training. It's monitored constantly. As I told you, it's monitored not only by the sponsors, but in most clinical trials, I'll have an independent committee we don't even know who these people are. They're blinded from the investigators that will be looking at data, and they'll be looking in both directions. There was recently something called the Cabinet trial, which looked at a drug cabozantinib in neuroendocrine tumors, and it was stopped prematurely. And the reason it was stopped prematurely is because they found the drug was so effective that, in fact, it would be unethical to continue the trial and randomize people to not receive it. And so the trial was stopped. On the other hand, there's some trials that can be stopped because of futility. It means that there's no chance this drug's going to work. By early results, we can already tell that this isn't going to work. So going to the end of the trial wouldn't be ethical to those patients. And sometimes trials are stopped due to safety reasons. Side effects must be reported. And if you ever participated in a clinical trial or have participated, you'll know you get asked 1,000,000 times about every side effect you have. It's monitored, it's reported. As an investigator, I sign off on every single one, and we send them off, and we make sure that we're collecting every side effect to ensure that if our patients are having side effects, we can monitor it on the whole trial. And all clinical trials must be reviewed by an independent research ethics board called an REB. The REB could be of different capacities. Sometimes it's a hospital level, a university level, different organizations, but it has to be reviewed and approved by an ethics board to ensure that all these principles are being met. So as I mentioned, clinical trials can take many different forms. People think it's really just testing of drugs, but it can be much more. They can be done anywhere along the cancer journey here. They can go right from their prevention, screening, diagnosis, treatment, survivorship, and end-of-life care. There can be clinical trials in all those areas that can help look at people preventing a disease, detecting it earlier, improving the outcome of disease, making life better, so symptom control for people with the disease, and prevent the disease from coming back, and that's like adjuvant therapy or preventative therapy. So there's really 4 phases of a clinical trial, and if you're thinking about participating or you get an option, it's important you understand what phase of clinical trial it is, because they're very different depending on the phase, and the endpoint is very different. So phase 1 clinical trials really are the first clinical trials often that it's being done in humans. They're there to evaluate safety, to determine the dosage. So often a phase 1 clinical trial will start at a lower dose and then for 5 patients and move up for the next 5 patients to try to find out what the dosage is in humans and to identify side effects. Now, generally, we do get efficacy. We do get some efficacy data to decide to move it on to phase 2 or not. But the primary goal of phase 1 trials is to really understand the safety of the drug. Generally, phase 1 trials are for patients who've exhausted all conventional options, so there isn't any other conventional options for them. Phase 2 trials, I like to think of it as a thumbs up, thumbs down, and that is really the first test of effectiveness. It does not prove efficacy, and that's very important. Phase 2 trials, although they're testing a drug against something else, let's say, they don't actually prove efficacy. They're there to say, is there enough of a signal to move forward in this regard? Phase 3 trials are the groundbreaking trials. That's what we hear about. These are the trials that confirm effectiveness, monitor side effects, and really decide whether this treatment is to be the new standard or should be considered a new standard in the treatment. And then the final part is phase 4. Phase 4 clinical trials are what goes on every day. After a drug is, let's say if we're talking about a drug, after a drug is approved and available and people are being treated on it, we have to monitor it in the population, because a clinical trial is a very nuanced and a very regulated environment. Sometimes when things happen in the population, either side effects come out that we haven't heard of before, or we understand that maybe it doesn't work as well in the population as it did in this narrow clinical trial. So again, phase 1 clinical trials could be the first test in humans. They're generally small trials, and the goal is safety. We're trying to understand the side effects, understand the correct drug dosage, and we're also looking at efficacy as well to get a larger picture. But this is a broad stroke in terms of efficacy. Often disease agnostic. That means that it could be, for example, there's colon cancer patients, neuroendocrine cancer patients, breast cancer patients, all within the same clinical trial. And they're not available everywhere. Phase 1 centers, as you can imagine, looking at these outcomes, have to be very highly regulated and monitored. And so it's usually at sort of highly specialized academic centers. And these trials take place over a time period of months. They're usually fast and rapid, and often a clinical trial will open. Maybe we're opening to 20 patients. Those 20 patients get accrued, and then the trial closes. Phase 2 clinical trials also have usually limited patients, but are higher. They could be 100, maybe a couple hundred at max. But really, here the emphasis is on effectiveness. So we understand the safety, we understand the dosage. We want to get a picture here to say, does this drug seem to be working or does it not seem to be working? So we've now switched from safety to efficacy. Is this worth moving forward? Is the time, the money, and also the patient's well-being, all of that, does it make sense to move this drug forward or to move this therapy forward? Usually at this point, it's going to be disease-specific. Of course, we're going to gather more information on safety, and phase 2 clinical trials can take years rather than months. So this is, again, the warning is that sometimes we overinterpret phase 2 trials. We get excited. We see a phase 2 trial and we say, wow, this is a positive result. Oh, this is the new treatment that should be out there. Phase 2 trials aren't designed for that. They're really designed to give a thumbs up, thumbs down to move forward. And sometimes, this was a front page of Time magazine about, VEGF inhibition or a drug called Avastin, which sometimes there was a lot of enthusiasm about it at the phase 2 level, but at the phase 3 level, although it was a positive trial, it again didn't show the widespread benefit for colon cancer that many people thought it would. So the phase 3 clinical trial is pivotal. This is where we really take all the science, implement it, and say, is it effective? How effective is it? And we start to look at things like how many people need to be treated to get a positive result? What is the extent of the benefit? So when we're talking about benefit, there could be, as you heard with Lutathera, a very strong benefit. There's other drugs where you have to treat hundreds of people just to get one person to respond. So we want to be able to understand that, right? This can involve at least hundreds, often many thousands, and even tens of thousands of patients in large trials like cardiology, et cetera. It's randomized. I'm going to talk about that in just a second. And it doesn't necessarily mean it's one drug. It could be a combination of drugs. It could be drug A and B versus drug A. It could be drug A and B versus drug B and C. It could be A followed by B, just by A. So you can often get very complex designs here. We're really trying to get all the information here because this is the final step before approval of a new treatment. So what is randomization? I think this really scares people sometimes when we talk about the topic of randomization. It is basically what we say. It's a randomized assignment of which treatment you're going to get. It can be blinded or unblinded. So often if the treatments look alike or can be, we may not know. You as a patient may not know which treatment you're getting. I as an investigator may not know which treatment you are getting. Sometimes if it's a treatment, let's say a trial of like surgery versus a drug, or if it's a drug that's given as an injection versus a drug given as a pill, it's unblinded because it's impossible to blind it. Randomization doesn't need to be 1-to-1. So if we have a strong suspicion a drug works, and often we do something called a 2-to-1 or a 3-to-1 randomization, where the trial's designed that 2 out of every 3 people would potentially get randomized to one of the arms. And this is where we account for bias. So there's a lot of things called confounders. So things, for example, some people who are younger and healthier may do better on a drug than some people who are not. Some people have a certain type of cancer. For neuroendocrine, some people who have a pancreatic neuroendocrine cancer may do better on a drug than some people who have a lung neuroendocrine cancer, for example. Or there may be other factors. People with diabetes may change the outcome of this drug on their cancer. And so this is why we do randomization. We're trying to reduce bias. You have a random chance of being in either arm, so a lot of those confounders should randomize out. They should be in both arms. Sometimes there's factors such as like the site of disease or other factors which really come into play to the biology, and therefore we stratify. We make sure that we don't allow that bias to be introduced into the trial, and that affects the randomization. So this is what randomization looks like. Sometimes it's a bit more complex than this, but really you have a group of patients. They're randomized usually in a way that is electronic to a treatment group or a control group. And control group could mean the way we treat the treatment today. And then they're followed up. And then we follow up to see who had a benefit from the treatment or not. And then statistical analysis is done to compare the results with a high degree of efficacy. The standard that's used is much like we see if you see on polling or you see on other things. It's a chance of 19 times out of 20 that this result is in fact valid. So again, then this is how things go in terms of approvals. It's usually post-phase 3, which is why the phase 3 is so important. Sometimes when there's no other treatments available and there's really promising results in phase 2, as I talked about, there is approval post-phase 2, but usually it's with conditions. And that usually means, yes, we don't want to deny patients this treatment, which looks very, very promising, but we recognize it's not definitive yet, and therefore you have to either do a phase 3, but we'll allow patients to start getting treated with it, or there's other data collection that's required. Phase 4, as I mentioned, is after your health authority approval. This monitors drug effectiveness in the population. And so people sort of ask, well, why would it be different? You've already done this in these very large, very expensive, very resource-intensive phase 3s. Well, that's because real life is different than a clinical trial. Clinical trial, you have a number of people monitoring and checking and doing all things. Sometimes when you actually, what we call real-world experience, you release the treatment into the population, things are a bit different. And maybe things don't work on the population as much as we think it is. Study populations are fairly select. Not everybody, unfortunately, we're trying to improve this, and a lot of work needs to be done, but there is equity issues in clinical trials. For example, people who don't speak English, or people who come from rural areas. It may not be practical for them to participate in a clinical trial because of the visits and for the amount of time that is required. So those people maybe aren't reflected. And one of the problems we still have in clinical trials is age. We sometimes clinical trials are restricted to age to reduce risk. But when we go into population, we don't always judge people by their age. We judge people based on their viewpoints, their experience in life, how they want to be treated. And also, many people are a candidate for a clinical trial at 75, and many people are not at 65. So the patient population of a clinical trial isn't often exactly parallel to what we see in the population. And then there's rare side effects. You know, there's a drug called Celebrex, for example, that passed phase 3 clinical trials, but then they found some cardiovascular issues when it was done in a population level. Because when you treat with millions of people, rare side effects come about. The COVID vaccine was an example, right? We saw that there was a COVID clinical trials, but when we did the population level, there was a very low risk, but a risk of DVTs, right? Pulmonary embolisms. And so that can often be only detected, these very rare side effects only be detected when you're looking at millions of people rather than thousands or even tens of thousands. Or a period of time. So much like you heard today about the radiopharmaceutical trials, there's only follow-up for a certain period of time, and there's a rate of hematological toxicity, or MDS, and even leukemias. But we also need to understand what happens 5 years down the road. Sometimes the side effects don't manifest themselves till way later after the clinical trial is over. So that's why these phase 4 trials are important. Another big question is who does clinical trials? There's always this sort of this idea of that there's this clinical trial, but who's behind it? Is some evil pharma that's doing it? You know, and there are a lot of drug company trials, and those are called pharma trials. And, you know, to be honest, yes, they're trying to advance their drug, they're trying to make money, they're trying to build a new product that they're going to put on their market. That's the reality of the situation. That being said, a lot of big advances, like you just saw about radiotherapy, are done by pharmaceutical companies, and they often have the resources to do these very large trials. It is still regulated the same way. There is no difference in how we regulate clinical trials despite who the sponsor is. Those high ethical standards and those safety standards are met regardless of who's the sponsor. There's academic trials, like the Cabinet trial I told you was an academic trial. Academic trials often answer questions that pharma wouldn't. So maybe that are not going to lead to a new therapy or are not going to lead to a drug that's going to make more money, but potentially are going to ask questions that are still very important to patient care. Unfortunately, these trials can be difficult to do for funding reasons. Trials are very expensive to do due to the high standards we maintain, and they can be very difficult to get funding for. And often, if an institution, when you're looking at an academic trial, your institution, you're going to lose money on it. And that is the institution is going to have to supplement these areas because academic trials often don't pay for themselves. So this is why research, advocacy, fundraising is so important. Who approves clinical trials? So, you know, they be done in the US through the FDA. In Canada, Health Canada has to approve a clinical trial before it can go anywhere. And the questions they're going to be looking for is the drug being used in the right way for patients being studied to ensure that this is a rational approach, makes sense, we're actually trying to improve patient care here. Are the risks being lowered as much as possible? Are we doing everything we can to manage any of the risks that involve of a clinical trial? Are the best interests of the participants being taken into account? That is paramount in any clinical trial. No matter how spectacular a drug may be, et cetera, no trial can be approved unless every effort is made to keep patients at the center. And can the goal of the trial be met? Is it feasible? To a lesser extent, that's really our internal responsibility, but the regulatory authorities still look at that. So why are clinical trials so hard to do? Well, they take a long time, often many years. Often 8-10 years is not an uncommon timeline for a clinical trial. To collect this amount of patients, to collect the data, the funding, all the work that requires is fairly huge. And clinical trials, especially phase 3 clinical trials, are very expensive. We're talking tens to hundreds of millions of dollars to do a single phase 3 clinical trial. Often they're not accessible to everyone, as I outlined, and that's a problem that we're trying to address. Eligibility can be too restrictive, so there's a balance, right? When you talk about safety, you want to make sure that you've taken every risk to increase safety. So you're going to say anybody who has a lab value over X, anybody who has any certain characteristics, you can't participate in a clinical trial because your risk may go up even a little bit. On the other hand, when we release to the population or we think about how we actually want to treat patients, one lab value isn't going to stop, right, our desire to improve our patient outcomes. So it's a fine balance we're trying to find here. Sometimes there's low accrual on trials. We had a trial in the neuroendocrine community looking at lung neuroendocrine tumors, a very important trial looking at somatostatin analogs in lung neuroendocrine tumors, and unfortunately it didn't accrue well. And so the trial had to be closed. And unfortunately in Canada, the result of that is we don't have funding for somatostatin analogs for lung neuroendocrine tumors because the trial couldn't complete. And so the end result of this unfortunate low accrual was that we now can't get funding for that drug because the data isn't there to get the funding. And then, as I mentioned, there's a lot of misconceptions. Some people just do not want to participate in the clinical trial, and fine enough, that's just not right for them, but a lot of people feel that they're a guinea pig and they're worried that they're being exploited. The other thing to keep in mind is that sometimes there is an expense to patients, and there are some expenses that can be mitigated and some that aren't. You know, there's more visits on a clinical trial. That may involve, for example, more caregiver support. That may involve gas. That may involve, we try to mitigate things like parking and transportation, but there's fine lines as well, as you can imagine, about financial incentives on a trial. It may mean people have to miss more time away from work, let's say, until you have more visits, and that may prohibit them from being part of a clinical trial. And then the idea of placebo. Not all clinical trials involve placebo. Some do, and some very important advances happen using placebo. That's how we are able to prove that a drug works versus not. But people hate that word, placebo. So I'll give you a quick story. This is not actually Rod, but I'm a huge Edmonton Oilers fan, even after last night. This is a 67-year-old gentleman who had high blood pressure, controlled with pills, otherwise healthy. He's an accountant, he's married, he has 3 children and 6 grandchildren. He noticed some pain and bloating, and his family doctor ordered a CT scan. He was diagnosed with a small bowel neuroendocrine tumor that had unfortunately spread to the liver. In neuroendocrine tumor, it's low, intermediate, or high grade, and he was intermediate grade. So what was going through Ron's head at this point? The first thing everybody thinks when they get diagnosed with the C word is, am I going to die? What's my quality of life going to be? We have a lot of preconceived notions about cancer and what it entails in the cancer journey. And how can I fight this? What are my next steps here? What am I going to do? So Rod meets his oncologist and he hears about a standard treatment. It's an injection once a month, a big needle that goes in your butt. It's tried and tested, lots of studies have been done, and it's the standard of care. But Rod, you have an option. There is a new clinical trial looking at a newer class of drugs. The benefit is it's a smaller needle. It may be a better treatment for you, and it may be a better experience from this large needle that you'd be taking every month for maybe many years. So what does Rod start to think? Again, there's things that come into his mind. I'm not a lab rat. What if this treatment doesn't work? Am I putting myself a step back by participating on a clinical trial when there is a standard treatment? Is this pharma trying to make money off my bad fortune? Clinical trials are a sham. This is just a money-making phenomena, and I don't want a sugar pill, right? I don't want a placebo. These are the first things when you hear clinical trial. Many questions come along this. So this was the clinical trial that Rod was enrolled in. It's a trial going on. It's been completed enrollment, but we're waiting for the results. It's called Sorrento. It's a randomized, multi-center, open label, active phase 3 trial to assess the efficacy and safety of a drug called CAM2029 versus the standard treatment, which could be octreotide or lanreotide, in patients with gastroenteropancreatic neuroendocrine tumor. So this was a randomized trial. It's a phase 3 trial. It was multi-center, open in 16 countries around the world, and it was open label because the needles were different and we couldn't tell the difference. And in fact, we actually had patient involvement on this trial when we designed it. Josh is part of our steering committee on this trial and is involved in sort of understanding the patient perspective on a clinical trial. So what is the rationale for doing a clinical trial in this scenario? Well, these higher or shorter intervals between somatostatin injections in these patients have been shown to actually help symptoms and in fact maybe help tumor growth. But a lot of this data has been what we call retrospective. It's been like, okay, well, someone tried a high dose and we can collect the data afterwards and look, it seemed to work. But as I mentioned to you, there's a lot of biases built in that. Maybe the reason they chose the high dose because the patient was healthier to start off with. Maybe there's other factors that were involved. So far, we didn't see a lot of increased toxicity, but again, it's never been tested in a scientifically sound, scientific methodological way. It's all been just experience-based design. So the end result of the conclusion is prospective trials are needed. You hear this a lot at medical meetings. Prospective trials are needed. We think it's promising, but prospective trials are needed, meaning we need to actually do the trials to move forward. So this was the trial design of this study. Patients were randomized, so it's 300 patients on this phase 3 study. These patients are randomized in a 1-to-1 fashion to receive the new treatment, CAM2029, every 2 weeks, or the standard treatment, either octreotide or lanreotide, every 4 weeks. So first thing, there's no sugar pill. You're guaranteed to get the standard treatment you would have got anyway. Okay, that's the first thing that we talk about when we talked about this trial. The second thing is you have a 50% chance of getting the new agent. Okay, well, what if you don't get the new agent? What's the advantage of then being on the clinical trial? The advantage is there's something called crossover, which means that if the standard treatment doesn't work for you, you can cross over and you'll get the new treatment. Okay, so there's an advantage to the patients there to participate. For example, with this trial, though, in Canada, I can tell you what was a disadvantage. Well, the disadvantage was these standard drugs in Canada are often given at home. We have the drug companies supply a nurse to go to the home and give the drug every month in the home. And it's very convenient for people. The disadvantage to participating on this clinical trial was that was no longer possible because it's a clinical trial and we have to monitor everything. Even those standard treatments had to be done in the hospital, which meant patients had to come every month, and I still have patients who are on this trial, had to come every month into the hospital to get the same injection they would get at home if they didn't participate in a clinical trial. So that is a mitigating factor. It made it hard. I had patients, I see patients from right across this province. I had patients from hours away who come to see me, and they wanted to participate, but even with parking support, even with some transportation support, it just wasn't feasible, to think in the month of February when we have, 30 centimeters of snow that they wanted they're going to be able to get in. And so unfortunately, they couldn't participate. So I'm not going to go through these, but there was a lot of inclusion criteria. We had a number of patients, for example, yes, I want to participate in a clinical trial. We talk about it, they sign consent, and then we find that there is something that meets an exclusion criteria. A lab value doesn't meet, or their scans don't show that we have a measurable lesion. The lesions have to be measurable. Sometimes surprises happen, and unfortunately, that's called a screen failure, which means that we have failed to be able to put that patient on the trial. And often it's because of these strict regulations we have around safety that unfortunately limit it. So what should Rod do? Yes, no, does he need more information? He asked lots of questions, and I think that's very appropriate. I actually worry more when patients say, yes, I'll participate. And I'm like, whoa, hold on. Don't you want to know about this? These are questions you should ask. I want you to feel comfortable with this decision. So he asked a lot of questions. He realized there's been a lot of science research and study to reach this point. This isn't somebody, sitting at a bar at 2 in the morning on the back of a cocktail napkin writing down some ideas. This has been years of work to get here. He thinks about how the trial may benefit him, but again, he thinks about others, and he thinks about other people, about how it may benefit. He talks to his family, the support network people have. Important for them to be part of that decision because people have doubts, and he decides to participate. And one thing he does find out is he gets a little bit of a concierge service, and I think this is common with clinical trials, is if you're on a clinical trial, you have so many people monitoring that you actually have probably a little bit more of an engaged team and a resource-intensive team than you do if you don't participate in clinical trials. So what should people think about? And hopefully we've covered all these topics when you're thinking about a clinical trial. What are the benefits and the risks? What is the purpose of this trial? Is this a brand new treatment, which sometimes it is, or has it been tested before? Will this improve my health today, or will it be down the road? And we were participated in a clinical trial that looked at a drug called everolimus, but we wanted to try to understand the factors that would decipher between people who responded and didn't respond. And what it entailed was an extra biopsy, which isn't a small thing, right? It's a needle being put into your tumor, and there's risks involved. And so the reality is it wasn't going to help those patients. It was going to help patients down the road. And I have to say, I had nobody who we talked about in the trial say no. They all wanted to participate. They all were willing to go through the extra biopsy because they specifically had the idea that if this is going to help people down the road, I want to be part of that. Who is the sponsor? Is this a pharma trial? Is this an academic trial? And again, I'm not saying a pharma trial is bad or an academic trial is good, but knowing the sponsor is important. What phase of trial is this? That's very important. Is this the first trials where we're looking at safety? Is this like efficacy trials? Is this a phase 2 trial? What are the known side effects that we know? And I think it's fair to, people always ask me, well, is this going to work? Well, I don't know. If I knew that this would work, then I couldn't be offering this trial. That would be unethical, right? But why is there enough enthusiasm for this trial to go forward that somebody is investing all the time and resources and money for that? What is the control arm? Is it a placebo control arm? Which at times makes sense. And I'm not against placebo-controlled trials, but not all trials are placebo-controlled. What is the current standard of care? So what would we be doing now? If not for this trial, what would we be doing, as I outlined in that previous example? What is the follow-up? There's a lot more follow-up often on clinical trials. Some people like that. On this trial, for example, there was more CT scans that would be done. Some people like that. Some people like having more follow-up. And some people, that's a deal breaker for participating in a clinical trial. What do I need to do? Sometimes it's coming to more visits. Is it more scans? Is it monitoring my side effects every morning in electronic diary? What is my investment in this? And why is this trial good for me? Why as my healthcare provider, are you recommending or talking about this trial with me? And again, it's really important that what normally happens is somebody in the circle of care introduces the trial, and then you want someone independent to explain the trial to the patient, so that they have an opportunity to be able to ask questions in a non-judgmental fashion. So I'll say clinical trials aren't easy, but they're very important. We're here today because clinical trials are continuing to move the needle forward in these cancers and many other types of cancers. And with that, I'll stop and take any questions I can add.
Thank you, Dr. Singh. I think we do have time for maybe one or two questions. I do have one from online. So there is a person watching on YouTube she says that she's a 20-year survivor of a phase 2 clinical trial with a 67% success rate at the 5-year mark, 55% success rate at the 10-year mark. She hasn't seen the 15-year mark rate yet, and the doctor who started the trial is now retired. So her question is, for how many years after a trial does the trial keep track of the outcome?
It's a really good question. The question is, how long do we monitor the trial? There is a certain period of time, what we call a primary endpoint, which is how the trial is designed, and that could vary depending on, the amount of what's happening on the trial. Once that's been reported, most trials will report at least 5 years, but the data we collect, and the data has to be kept for, in Canada, at least 30 years of time. So there should be data, but often after some period of time, they will stop collecting the data and reporting on it. But the information is kept for 30 years.
So most clinical trials are North American-wide or global, and in there we see FDA approval. Why does it take so long for Health Canada to then catch up? And secondly, have they ever unapproved or disapproved anything FDA have approved?
Yeah. Yeah, so some trials are global and some trials are in Canada only, and some trials can be one center only, something called, there's some trials called IITs or investigator-initiated trials, which might be only in that one center. Some could be province-wide, some could be national. I would have to say, FDA normally, it's a 30-day time period. So when you submit a clinical trial to Health Canada, you submit a clinical trial to Health Canada, it usually takes them they have 30 days to approve or to ask questions about the trial. So the time period of Health Canada isn't so bad. I think the delay sometimes with clinical trials is really the sponsor. You know, we have to have a separate sponsor in Canada, so it's not always as simple as because, you know, the FDA has approved it, you know, we need to have a sponsor within Canada, and that can sometimes be the challenge. You know, we have to have somebody hold the clinical trials, what they call the clinical trial agreement in Canada. So if we're participating on the United States on a very large level on a trial, there's one sponsor in the United States who would hold the clinical trial agreement. Bringing that trial to Canada, even into 2 sites, we have to have our own Canadian clinical trial agreement holder, and that's primarily the delay. Yeah.
I can shape this question several ways, but in a clinical...
Shape it the easiest way possible.
Yeah, okay.
Yeah.
In a phase 3 setting, does the length of time before readout impact like downstream overall survival and median survival rates in terms of extrapolating? If you've got a 10-year window, for example, of that clinical trial before readout, the assumption would be, if you're tracking positively, that you're going to have 5-year overall survival rates and so forth be positive. So if you're running a 2-year trial, for example, and then you get the readout, you're going to have to think about what's going to happen to the 5 and 10-year numbers. So length of trial does have an impact in how you measure the OS and ME?
So yes and no. Every clinical trial has what we call a primary endpoint. That could be overall survival, but it's specified, you know, or it could be progression-free survival, which means slowing the tumor growth. And that the clinical trial is read out at the endpoint. Now, depending on how quickly these things called events, is when somebody has progression or doesn't have progression happen, the endpoint could vary in between, but it's always going to be designed around that endpoint. And what happens is, for overall survival, at a certain time period, when you look, and the stats are designed around that specific endpoint. So at a certain time period, it's not necessarily a 5-year cutoff or a 10-year cutoff. It's just at the time period where the stats show with that degree of certainty that there is an improvement in overall survival in these patients, that's when the readout happens. That's why we never really know when it, you know, I can't say this trial is going to read out on April 1st, 2026. It could be a year before, it could be a year after. It sort of varies depending on how effective the drug is or effective it's not. For example, a trial that's very non-effective, unfortunately, will read out very early because it will show that the stats are not going to work here. We've already proven it's not working, or the opposite, right? So it sort of depends on those factors. Okay, well, thanks so much. I think your lunch is here, so thanks so much for having me. Thanks.
Thank you, Dr. Singh. So quick couple of